resistance is a global public health problem. The number of deaths due to untreatable bacterial infections rises every year, and more than 1/3 of that deaths are caused by Escherichia coli or E. coli and Klebsiella pneumonia, bacteria responsible for most of the hospital-acquired infections that cannot be prevented because there are no vaccines available, even though its development has been set as a top priority by the World Health Organization since 2017. But all the vaccines tested until now in clinical trials were based in the same strategy, to induce antibodies against surface epitopes of bacteria. And despite their immunogenicity, they do not promote a reduction in the rate of infections. This is in accordance with the growing evidence showing that antibodies against the surface of bacteria are not protective, which suggests that these bacteria share a mechanism of immunosuppression that blocks our immune system. A mechanism that we have been exploring at ImmuneTap, and that is based on the excretion of a protein, the GAPDH. We now know that upon infection, E. coli and Klebsiella pneumonia release this GAPDH that promotes an early and fast production of IL-10, that in high concentrations leads to an overall immunosuppression that creates the optimal scenario so bacteria can proliferate, which can culminate in a generalized and uncontrolled infection. Studies in murine models showed us that when we induce antibodies against GAPDH, we prevent this immunosuppression and the immune system became able to control the infection. Now, we are performing an observational case control study to evaluate the levels of IL-10 and antibodies against GAPDH in the blood of individuals diagnosed with E. coli or Klebsiella pneumonia bacteremia and in the matched healthy controls. With this study, we aim to answer the question, can antibodies against GAPDH protect humans from infections caused by E. coli and Klebsiella pneumonia? The results that we get until now are very interesting. IL-10, that represents a key step in the virulence mechanism of GAPDH, was detected in almost samples of infected individuals and in concentration significantly higher comparing with controls. On the other hand, the levels of antibodies against GAPDH were significantly lower in individuals infected both for E. coli and Klebsiella pneumonia comparing with controls. We were also able to associate these levels of antibodies against GAPDH with diagnosis of bacteremia. Using a model of logistic regression, we found that increasing the levels of antibodies against GAPDH reduces the likelihood of a person being diagnosed with a bacteremia. These are the first results in humans that strongly suggests that antibodies against GAPDH can prevent infections caused by bacteria that use GAPDH as a mechanism of virulence. More than that, we estimated a threshold of protection. By distributing the infected individuals according to the concentration of antibodies against GAPDH, we found that with concentrations of antibodies against GAPDH higher than 1 microgram per milliliter, we could prevent at least 90-60% of infections caused by E. coli and Klebsiella pneumonia. This is available information to evaluate the efficacy of a vaccine that we created at ImmuneTap named Paragon Novel Vaccine, a vaccine that induces the production of antibodies against GAPDH, antibodies that now we know are associated with a lower susceptibility to infections caused by E. coli and Klebsiella pneumonia.